So welcome to one of our lectures. Today we're going to be talking about Hardy-Weinberg. But I wanted to go ahead and list for you the big ideas and the questions. So today we're going to be covering what is Hardy-Weinberg, which I'll shorten by HW. When does it happen? And lastly, how to apply it. So how to apply Hardy-Weinberg. So this is going to be key, especially for next week. Our lab that we're going to be covering will be coming lab number 2. So if you want to look on the PDFs online, they're available there because we're going to be working on this in lab number 2. So let's get started on what Hardy-Weinberg is and how to apply it. So what is the Hardy-Weinberg principle? Hardy-Weinberg principle is the statement for what happens when there's no evolution going on. So no evolution. So that basically means there's no selection. There's no selection occurring in your population. So you look for 5 things to make sure there's no selection. And what are those 5 things? If there is selection going on, you want to have a small population. So to fulfill Hardy-Weinberg, you need a large population, you need random mating, you need no gene flow. So going back to last week's lecture, there's no gene flow, so no migration or immigration. Large population, random mating, no gene flow. We're also looking for 2 other components that basically say there's no selection going on in your population. So competition and no mutations. So these are the 5 things you need to fulfill if you're having Hardy-Weinberg occur in your population. All right. Now we're going to cover what the equation is for Hardy-Weinberg and why you would use it. So in a population that has no gene selection, or no gene flow, no random mating, it's a large population, you're tending to look at a neutral trait. So this could be a trait such as a T or an A or G or C. Something random that you're looking in your population. Something that doesn't affect whether or not you get eaten by a hawk. So in the case of the stickleback fish, that means you couldn't use the spikes. You would have to use something else that doesn't apply to selection. So what is the equation for Hardy-Weinberg? It's P plus Q equals 1, where P and Q represent allele frequencies for a trait. And you have P squared plus 2PQ plus Q squared equals 1. So this is your population. So these represent your homozygotes. 2PQ would represent your heterozygotes. So in this case, we'll say P equals your dominants, Q equals your recessives. So P squared would be the fact that you would be dominant. 2PQ would represent the fact that you are big A, little a, or A, big A, and Q squared has to do with the frequency if you were little a, little a. So that's the equation explained. So what? How do you use it? How does it work? So now you have your trait. Let's say we can look at something like tongue curl. Tongue curl, can you curl your tongue or not? In humans, it doesn't really have selection. Just because I can curl my tongue doesn't mean I can get more water and outcompete you and now have more kids. Remember, fitness is defined by the number of kids you can have and your children, your offspring. So let's look at tongue curl. So we'll use T for dominant, little t for recessive. So I believe tongue curl is dominant. If I'm incorrect, I'm just going to use this for this example. And I'll go back and fix it next time, but just to walk us through, we'll say in our population, tongue curl occurs 20% of the time. So it has a frequency, the frequency of capital T is 0.2, 20%. So therefore, we know the frequency of little t has got to be 0.8. So from here, we can predict not only the number of individuals that would be heterozygote, we can determine their phenotypes, and we can also determine how many would be in the population. So this kind of goes back to the stickleback. You had your Punnett squares, and you could predict what traits they had, and you could also look at why they have those traits because of the Punnett squares. So here's a mathematical model. Remember, modeling is one of those key skills that AP Bio wants you to have, the College Board. So we're going to make a mathematical model of this population that has the tongue curl. So we know the frequency is TT, so now we can figure out how many individuals have it. So 0.2 squared, 4% of the individuals are going to have this. So in our class we have 11 students. You can predict if we were a significantly large population, maybe the whole school would have this trait. You could also look at how many individuals would be heterozygotes. So we can plug that in, and lastly q squared is equal to 0.8, 0.64. So most of the individuals in our population are going to be recessive. They're not going to have a tongue curl. So this is where we take data and we're going to draw conclusions. And it's a lot of just mathematical manipulation and knowing what these values mean. So here, you can use a percentage to figure out the population, building from the frequency. You can also work backwards, and I'll show you that in the next example. We can also work backwards, so knowing the frequency of dominant phenotypes, you can work backwards from there to find out what your frequency of the gene is. So I can use this to find out how much of big A I have and how much of little a I have. So knowing that the dominant phenotype is 0.8, you can use that. You know that the dominant phenotype is going to be both the big A, the homozygotes, plus the heterozygotes. So using that information, you know that 0.8 is equal to p squared plus 2pq. So you can plug this into the equation, p squared plus 2pq plus q squared is equal to 1, and you will find that q squared is equal to 0.2. So I used 0.8 over here, subtracted from both sides, shortcut down to q squared equal to 2. Take the square root of both sides, the square root of 0.2 would give you q. From there, then you can work your way back to p and find out if an equation is in equilibrium or not. Again, this is just algebra, working back and forth between different values. So being able to manipulate that equation will help you a lot in trying to figure out exactly what you need. Alright, so now I'm going to work through some of the problems you got on this worksheet, the one that's the Hardy-Weinberg practice problems. So I'll start with number 1. So we have something to show your work on. So you have sample the population which know the percentage of homozygous recessive genotype, little a is 36%. So calculate the following. So starting with little a, we know that little a is recessive. That's going to be equal to q squared. So from q squared is 36%. So 0.36 equals q squared. Therefore, q, take the square root of both sides. Remember, you can use a 4-function calculator. Of q squared is going to be 0.6. So using the equation p plus q equals 1, you can find that p equals 0.4. Now you know the frequency. So these values represent the frequency of the allele, not necessarily the frequency of the genotype. So frequency of the genotype little a is here. The frequency of the allele is here. The allele is just the frequency of q. All this is the genotype. So that covers A, B, and C. Moving on to D is the frequencies of the actual genotype. So to get the frequency of p squared, we give you the dominant, we get 0.16. And to get the frequencies of the genotypes that were heterozygotes, the big A, little a, you have to use again 2pq. 2 times p, 0.4, times q, 0.6. You end up with 2 times 0.24 0.48. So if you look at your population, this would give you some numbers. This would give you 48% of the population was heterozygotes. 16% of the population would be your dominant, and 36% of your population would be recessive. So that's working through number one. So that was one format of working around with the equation. You could also use Hardy-Weinberg to show if a population is evolving or not. So this is kind of goes into that same kind of logic that was going on with the chi-square. You use it to show that something isn't, to prove that it is usable. So following that same logic, we know that Hardy-Weinberg shows when a population is not evolving. So if you can show that Hardy-Weinberg is not true, you don't get this p squared, q squared, 2pq perfectness, then you can show that population is evolving. Again, you could also show a population is evolving by eliminating 5 of those traits, but if you're not sure about it, then you can use this mathematical model to prove that something is evolving by proving Hardy-Weinberg wrong. So where is a problem like that? If you go to number 13 on the back of this Hardy-Weinberg practice problems, that's an example of that. So it says for a particular gene, assume a population has a frequency of heterozygotes at 10%. A study of this population shows that homozygous recessive is 16%. Is it at equilibrium? If it is at equilibrium, that means it is not evolving. If it is not at equilibrium, and we can show that this is false, then Hardy-Weinberg is not at play and the population is evolving. So how do we go about doing this? The frequency of heterozygotes is 10%. So this means 2pq equals 10%. We also know that the population has a frequency of homozygotes that are recessive at 16%. So q squared equals 0.16. From those two starting points, you can go from there to go through the rest. So q squared is 0.16, so we want to square root both sides. Q is equal to 0.4, therefore p is equal to 0.6. Knowing these values, we can go ahead and test it against 2pq and find out if that would give you the result of heterozygotes. So test heterozygotes, and you would test 2pq, 2 times 0.4 times 0.6, and you would find it is 2 times 0.24. You would expect, if it was Hardy-Weinberg in equilibrium, 0.48. So we check that back up against. So check 0.48 equal to 0.10. No, that does not apply. So because that does not apply, we can reject Hardy-Weinberg, and therefore, since we rejected Hardy-Weinberg, we know that the population is evolving. It is under some form of selective pressure where heterozygotes or homozygotes are being favored. Alright, so that concludes our section on Hardy-Weinberg, how you would apply the equation, why it's useful, and different ways that you might use the equation for Hardy-Weinberg. If you have any questions, please feel free to shoot me an email on Schooloop, text, or ask for some office hours. I'll always be free for you to help you out, brunch and lunch. You just got to come in. I'll see you soon. Take care.